Thanks for having me here. My name is Jace Webster. I'm a graduate student in the lab of Chris Marr here at Washington University in St. Louis. And today I'm going to be talking about our development of a pipeline for detecting somatic variants using cell-free DNA. And today I'm specifically going to be highlighting our work with structural variants. So the reason that we got interested in doing this was because we really wanted to be able to use the cell-free DNA to non-invasively monitor disease progression. And specifically, we were interested in prostate cancer. And so what that would look like is you have tumor cells that release DNA into the bloodstream, and then we're able to non-invasively collect that blood and perform sequencing and then detect those somatic variants. I have some of those clinical results here, although our clinical collaborators are actually going to be sharing that full story out in the poster session today. So if you check out poster 65, I definitely recommend looking at that. But as we began doing that analysis, we found that unlike SNV or copy number alteration tools, the methods for detecting structural variants in this context seem to be lacking. Specifically, we identified 3 tools that were either developed for detecting SVs in this context or had been benchmarked in this context by their authors. And each of them seemed to have some limitations, at least in our eyes, for our use case. Mainly, they were either limited by the size of SVs they were looking at or unable to report some of the fusion or the events that we wanted to look at. And as a result of this, many of the cell-free DNA studies that we looked at used either ad hoc or proprietary methods for their analysis. Many other papers used kind of generic SV calling tools, which seemed to work, but they were unable to point to any sort of benchmarking to suggest that those tools were appropriate in this context. So to kind of handle this for us, we devised PACT, which stands for Pipeline for Analysis of Circulating Tumor DNA, as our sort of analytical pipeline. And how that works is we begin with cell-free DNA and a matched control from the same individual and perform initial variant calling using lumpy, manta, and deli, which are all sort of generic SV calling tools. And then we create a consensus call set from those tools. And then we follow that with region-based filtering. So this is done in targeted sequencing panels. So we first confirm that the SVs we're looking at are associated with the regions we actually targeted with our panel. And then we eliminate variants that are associated with either low complexity or blacklisted regions, essentially regions of the genome that are known to introduce false positives for this. And then we perform genotyping. So we compare that cell-free DNA against the matched control from the same individual, and then we also compared it to a panel of healthy individuals to confirm that what we were looking at really seemed to be like a somatic cancer-associated SV. And then finally, we applied a read support filter to confirm that we saw both split read and paired read evidence of these calls. And the idea is that that should generate a list of high confidence calls for us. So we went ahead and implemented this and began to assess performance by applying it to a public cell-free DNA data set from a prostate cancer cohort with previously validated SVs that were validated in the matched tumor samples from these individuals. And so when we applied these tools to the cell-free DNA, we found that both PACT and Aperture, which is one of the other tools we looked at, were both able to recreate 100% of those findings. But we were particularly interested in what performance would look like at lower tumor DNA content levels. And so we performed an in silico simulation where we took public data from a prostate cohort and from a colorectal cancer cohort. And we performed a downsampling experiment where we combined that cell-free DNA or the tumor DNA data and combined that in silico with matched controls from each individual, respectively. And did that over 100 iterations to look at different tumor DNA content levels and found that our pipeline had superior sensitivity compared to the other tools that we looked at. And you'll notice in this data set and in the following ones, we didn't include SFICT because we actually couldn't get that tool to even work on these data sets. Finally, we looked at a public cell-free DNA reference data set that was generated by Horizon Discovery. And they report that there are 2 validated gene fusions in this data. Both of them are expected to have an allele frequency of about 5%. And we found that only PACT and Aperture were able to detect those. However, Aperture reported almost 1,600 SVs in this data set, and so there just seemed to be a lot of false positives from that tool. So based on all these results, just kind of as a summary, we feel that cell-free DNA is clinically useful, but the detection of SVs is sometimes overlooked in this context. But compared to those published tools that we looked at, SV detection using our pipeline had superior sensitivity in both the clinical cell-free DNA dataset and in silico simulations, and a superior false positive rate when looking at cell-free DNA reference data. So we believe that combining this pipeline with some of the more robust SNV and copy number tools that are made for this context, we're able to provide a more convenient and reproducible analysis for this clinically important analysis. And so with that, I'd just like to acknowledge the members of my lab as well as our clinical collaborators here at WashU. And again, we just point you to the clinician's poster outside that has their results from that analysis of prostate cancer patients, as well as my own poster. So thank you.